In this lecture, I'm going to talk about thyroid autoantibodies in Graves' disease. And as you may know that Graves' disease is an autoimmune disease. In it, the immune system makes very specific antibodies that stick on the thyroid and lead to release of too much thyroid hormone, which affect different parts of the body. And the most common cause of hyperthyroidism is Graves' disease, which is due to overproduction of thyroid hormone. And this overproduction of thyroid hormone may lead to thyroid toxic crisis, which is known also as thyroid storm, and it is due to acute complication of hyperthyroidism. So in this lecture, I'm going to talk about autoantibodies in Graves' disease. So some patients upregulate anti-thyroid stimulating hormone receptor antibodies, as can be seen here. These are TSH receptors. So some bodies or some individuals, they upregulate antibodies against these receptors. So these antibodies, they mimic the action of TSH and trigger secretion of T4 and T3 by the thyroid gland. So autoantibodies present in the serum of patients with Graves' disease include antibodies to TSH receptor, thyroid peroxisomes, and thyroglobulin. However, in this lecture, I'm going to focus on antibodies or autoantibodies to TSH receptors, as they are central to disease pathogenesis, and these include thyroid stimulating immunoglobulin or TSI, which is specific for Graves' disease and useful in monitoring Graves' outcome, and also long-acting thyroid stimulator or LATS. These antibodies actually bind to TSH receptor and mimics the action of thyroid stimulating hormone and may cause hyperthyroidism in newborn due to placental transfer or placental transmission. And also in this patient with hyperthyroidism or with Graves' disease, thyroid stimulating hormone binding inhibitor immunoglobulin may be detected, and these antibodies prevent thyroid stimulating hormone from binding to receptors on thyroid epithelial cells. So these inhibitory or inhibiting immunoglobulins are occasionally responsible for hypothyroidism in patients with Graves' disease. Thank you very much, Professor Ahmed Boulad.